Hello viewers, welcome to this particular lecture on photosynthetic dark phase. In this particular lecture, I shall be discussing the general process of photosynthesis and the different factors involved in photosynthesis, which include the Blackman's law of limiting factors, and then I shall be discussing the energy-rich compounds which are generated during the light phase, how they are utilized during the dark phase or the Kelvin cycle. And this particular Kelvin cycle is also known as the C3 cycle because the first stable photosynthetic product is a C3 compound, which is phosphoglyceric acid. And the overall process of photosynthesis clearly shows that there are 6 carbon dioxide molecule which is producing 1 molecule of sugar or glucose, but it is also producing 6 molecules of oxygen and 6 molecules of water. And here the starting compound water, there are 12 molecules of water which is utilized. So what we can see in the overall equation, there are excess of water molecules absorbed and some amount of that, that is 50% of that, is liberated out without getting utilized. So it is not a pure chemical process, it is basically a photochemical process. The site is the chloroplastid, but it occurs mostly in the green chlorophyllous tissue, and this is known as the photosynthetic apparatus. So the photosynthetic apparatus, if we follow the hierarchy, the leaf is the biggest structure which is easily available, and this leaf is having the mesophyll tissue. The mesophyll tissue is having the palisade and the spongy parenchyma, and within this particular parenchymatous cells there will be the chloroplastid, and within the chloroplastid there will be definite chlorophyll molecule. So we can very well understand that what is the efficiency of photosynthesis process, because each particular quantosome, which is the unit of thylakoid within the chloroplastid, contains 250 chlorophyll molecules. So basically the chlorophyll efficiency depends on the light available in photosynthesis. Now let us talk about the chloroplastid structure. The chloroplastid is a discoid structure having grana and intergranal region, but here we will be mainly considering the intergranal region, which is the stroma, and this stroma is basically the ground substance which is having all the enzymes required for the dark phase or the Kelvin cycle. Considering the absorption wavelength or absorption spectra of different pigments, we can clearly see that there are a specific pigment like chlorophyll A, which is having absorption more in the 700 region or 650 region. Chlorophyll B is having more absorption in the particular 400 to 500 nanometer region, and there are carotenoids which are slightly extended above 500. So what does it mean? Each of these pigments are basically complementing each other and making the process of photosynthesis more enriched and more efficient. There are different factors that affect photosynthesis that include the light intensity, and we know, as we have seen from the absorption spectra, the blue light and the red light are more important for photosynthesis, but it is the red light which is more important because it is absorbed by most of the pigment system. Considering the temperature is around 20 to 30 degrees centigrade. Above 30 and below 20, the photosynthetic rate may be reduced. Of course, water is important because they absorb excess of water. To what amount they are needed, they absorb excess and liberate out the excess water. Carbon dioxide concentration is so important because carbon dioxide is ultimately getting reduced to produce sugar. So definitely carbon dioxide increase will increase the process of photosynthesis. Oxygen concentration is having a negative role because oxygen is an end product. So more the oxygen liberated, lesser will be the rate of photosynthetic process. But a very unique graphical representation is there. Whenever this particular light intensity is increasing, the photosynthetic rate increases, but definitely after some time the photosynthetic rate becomes constant. Same is true for carbon dioxide. But for temperature, it so happens that if the temperature increases after certain time, the photosynthetic rate will fall. What does it mean? It means at higher temperature, the chlorophyll pigments and the machinery of photosynthesis is getting destroyed, as a result of which photosynthetic process is getting slower or lowered. Oxygen concentration is not showing any definite graph because what happens is oxygen initially will reduce or will increase because some of the enzymes will be activated. But if the oxygen is in excess, then there will be gradual reduction in the photosynthetic rate because of the oxygen poisoning effect. Considering a very important law which is called the Blackman's law of limiting factors. What does it state? It states that photosynthesis process actually helped by different factors like carbon dioxide, light, water, etc. But multiple factors, though it is determining photosynthesis, the actual photosynthetic rate is determined by the slowest factor. What does it mean? It means that the factor which is present in minimal amount will actually determine the rate of photosynthesis process. That means if say carbon dioxide is minimum, that carbon dioxide will be the determining factor determining the rate of photosynthesis. Then gradually the carbon dioxide increases and it is no longer the minimal factor. The next factor may be light. So now the light intensity will determine the rate of photosynthesis. And as the light intensity increases with daytime, gradually the light will no longer be the deciding factor, maybe the next factor is water. So in this way, Blackman gave a very important law which states that the minimal factor will always determine this process of photosynthesis, though actually it is determined by multiple factors. And coming to another very important phenomenon which is called the Blackman's reaction. Basically Blackman gave this reaction way back in 1905 when the internal structure of the plant or chloroplastid was not known, not even the chlorophyll structure. But he said that photosynthesis definitely had a photochemical reaction which is the light reaction, and there is a temperature sensitive reaction which is the dark reaction. And overall he also could explain the synthesis of sugar during the dark phase. So definitely Blackman that way is the scientist who for the first time denoted that photosynthesis is biphasic and the dark phase is independent of light and it actually produces sugar. But this particular process of photosynthesis, we say light phase and dark phase, but it is not actually right because light phase is fine, but it is better to say light dependent phase because without light this phase will not occur. But the dark phase incorrect is because that the dark phase is called light independent phase because it is not dependent on light, it is dependent on the energy rich compounds which is produced during the light phase. So we should say that the dark phase can operate in darkness. But you see, if the darkness continues, then ultimately what happens is this particular photosynthetic process or the synthesis of sugar will come down because there will be no energy rich compound that will support the dark phase. So it means definitely that there are very important energy rich compounds which are generated in light phase, that is in the grana or thylakoid, that is being utilized in the dark phase, and that ultimately produces sugar. And these compounds include the ATP, and that is produced by photophosphorylation, that is ADP is getting phosphorylated in presence of light, and at the same time H plus ion which is received by NADP will be reduced to NADPH, and thereby these two compounds or these two molecules will be utilized during dark phase during sugar synthesis. Let us talk about now the different aspects of the dark phase or the C3 cycle or the Kelvin cycle. This particular dark phase include a series of biochemical redox reaction that takes place in the stroma of chloroplastid in a photosynthetic organism. This particular cycle was discovered by Melvin Kelvin, James Basham and Andrew Benson in the University of California, Berkeley, and they actually utilized the radioactive C14 to indicate the actual process of photosynthetic dark phase. And this particular light independent reaction definitely utilizes ATP and NADPH as I have already stated, and it includes the most important phenomenon in photosynthesis, which is carbon dioxide fixation, and by this process the carbon dioxide will be reduced and thereby the global warming can be checked. So planting of trees can actually reduce global warming because it will reduce carbon dioxide, increase oxygen. The key enzyme for photosynthetic dark phase is called RUDP or ribulose diphosphate carboxylase and oxygenase. It is in pet name called Rubisco enzyme, and this particular Rubisco enzyme is having a very important phenomenon of carbon fixation, and there are different types of intermediate sugar which is produced, and ultimately this particular RUDP or ribulose biphosphate is regenerated. All these enzymes are present in the stroma and they carry out the process of photosynthesis smoothly. But as I told you, photosynthesis dark phase cannot continue for an eternal period in darkness because it is dependent on the light phase dependent energy rich compound, and if it is not there, then definitely this particular process will be standstill. So overall, the dark phase of photosynthesis include 3 or 4 major phases. Of course, there are innumerable number of phases which are present in this particular 4 major phase, and these phases include the carbon fixation, by which carbon dioxide is fixed, followed by the reduction of acid to aldehyde, and then followed by the hexose biosynthesis, and the other part of the carbon fixed will be regenerating RUDP back. So we call it in this way: carbon fixation, reduction, hexose biosynthesis and regeneration. These are the 4 major phases of photosynthesis dark phase. And the overall reaction of the Kelvin cycle can be like this: 3 CO2 plus 6 NADPH plus H plus 9 ATP giving rise to G3P or glyceraldehyde 3-phosphate plus 6 NADP plus 9 ADP plus 3 H2O plus 8 Pi, where Pi is representing inorganic phosphate. So the process of dark phase starts with carbon fixation, where RUDP is fixing carbon dioxide. It is producing an unstable intermediate compound and ultimately a stabilized compound called PGA. But when we consider the entire process, it is really very complicated, and it is Kelvin and his co-workers who categorically labeled the carbon and traced all these compounds, and ultimately when they proved this particular Kelvin cycle, which is the complete cycle of the Blackman's reaction, which was actually discovered by Blackman, Kelvin and his co-workers were awarded with Nobel Prize. So what I am trying to describe or what I am trying to explain right now is actually a Nobel laureate's work, and it is an immortal work which actually explains the synthesis of sugar in photosynthetic dark phase. The entire process, as I told you, has 4 major phases, but in fact there are 15 different steps which explain this photosynthetic dark phase, and I will try to explain these 15 steps. Step 1 is fixation of RUDP, and they will produce an intermediate compound which is actually carbon dioxide fixation by RUDP to produce particular intermediate which is N-diolate, and that ultimately will produce the compound which is the stable compound which is PGA. So first step is carbon fixation producing intermediate. The next step is actually producing the phosphoglyceric acid which is the stable compound. This particular 6 molecules will be generated, and these 6 molecules will be differentially broken down. So PGA or phosphoglyceric acid at the first step will produce another particular compound by taking up more ATP, that is 6 more ATP, to produce 1,3-diphosphoglyceric acid, and 1,3-diphosphoglyceric acid in step 5 will ultimately produce GAP or glyceraldehyde 3-phosphate 6 copies, and this is definitely by dehydrogenase, and in the process they will generate NADP and they will utilize NADPH. This 6 copies of GAP or glyceraldehyde 3-phosphate will be converted to different types of molecule, and we can see that one particular GAP will be producing ultimately one molecule of DHAP or dihydroxyacetone phosphate, and this dihydroxyacetone phosphate is actually an isomer of GAP, so it is also called isomerase enzyme action. And this DHAP or dihydroxyacetone phosphate along with another GAP in presence of aldolase enzyme will produce fructose 1,6-diphosphate, and this fructose 1,6-diphosphate is produced by the action of bisphosphatase enzyme, and they will produce fructose 6-phosphate. But the one particular GAP is taken out from the cycle, and this is utilized at a later time, and one particular GAP will be utilized in step 12 at a much later process. So what I am seeing is fructose 6-phosphate, which is an hexose sugar, will combine with one more GAP, and that ultimately under the action of trans-ketolase. Now we must understand, in fructose there are 6 carbon and in GAP there are 3 carbon. So there are 9 carbon altogether, and 9 carbon can give rise to a 4 carbon erythrose 4-phosphate and a 5 carbon xylulose 5-phosphate. This erythrose 4-phosphate will react with another one dihydroxyacetone phosphate to produce one 7 carbon compound which is called pseudoheptulose 1,7-biphosphate. And in the same way, this pseudoheptulose 1,7-biphosphate will be taken up by bisphosphatase enzyme to produce pseudoheptulose 7-phosphate. And this pseudoheptulose 7-phosphate ultimately will be producing one molecule of xylulose 5-phosphate and one molecule of ribose 5-phosphate. And this xylulose 5-phosphate will ultimately regenerate back RUDP, and this particular process is regeneration, whereas ribose 5-phosphate will also combine with it to regenerate RUDP. At the same time, there will be the phosphoribonuclease, which ultimately will regenerate the one molecule of ribulose 1,5-biphosphate that will re-synthesize or reproduce this entire Kelvin cycle. So what I am trying to say is that in this particular process, I have tried to explain that there is carbon fixation followed by reduction from acid to aldehyde or gap synthesis, and this gap is unequally distributed and reacted to produce different intermediate sugar which will ultimately regenerate RUDP, and thus this cycle will continue without any external help. Only that particular ATP and NADPH will be required to fix carbon. And in the process of photosynthesis, we can see that if there are 6 carbon fixed, then there will be actually 18 different ATP molecules utilized and 3 NADPH molecules required. So in this way, we can say that photosynthesis carbon fixation requires ATP. 1 carbon will require 3 ATP and 2 NADPH2, and thereby dark phase is not independent, it is dependent on the light phase. At least with the help of the energy, that particular dark phase will continue. So friends, what we have tried to discuss here is the entire process of photosynthesis, and I have given an overview, but I have actually stressed on this particular photosynthetic dark phase. In this particular Kelvin cycle, I have shown how the first stable photosynthetic product or phosphoglyceric acid is produced, and how this phosphoglyceric acid is converted to different other intermediates, getting reduced to produce GAP or glyceraldehyde 3-phosphate, and ultimately this glyceraldehyde 3-phosphate, part of it will produce hexose sugar, and the rest part will be regenerating RUDP to continue the Kelvin cycle process. But most importantly, the process of photosynthetic dark phase is dependent on light phase, and that is the ATP and NADPH which is produced during light phase. So without the light phase, the dark phase cannot continue. Thank you.